first speaker today is Dr. Davor Solter, and he is Emeritus Member and Director of the Max Planck Institute of Immunobiology and Epigenetics, visiting International Professor of the Suriraj Center of Excellence in Stem Cell Research in Thailand, as well as visiting professor at the University of Zagreb Medical School. And as well as discovering genomic imprinting, Dr. Solter is very well known for his work on teratocarcinomas and the origins of stem cells, mammalian cloning, and early embryo development. So please join me in giving a warm welcome to Dr. Solter.
Hi, thank you all for being here. It's really finding interesting. I was just watching a little, not so massive exodus of only very young people. I don't know whether this shows that science of optogenetics is a current science and imprinting is kind of something in the past. Actually, I will cover early days of imprinting to try to give you a feeling how it came about and how we were able to find it. It involves kind of somewhat fewer people than you just saw in the optogenetics field, but it's a different kind of science. Anyhow, but like everything else, it needed a technique to enable to go further, and the technique which we needed to develop, and when I say we, I mean Jim McGrath, who joined my lab in early 80s, and he was crucial in all these experiments which I will show you. And the technique was, how do we transfer nuclei in and out of mouse egg? Mouse egg is about 80 microns in diameter, but the pronuclei are somewhat like 15 nanometers, and if you want to take them out, you need a relatively thick needle, and if you put this thick needle into the cell, the egg will immediately die. So we had to devise a technology, which is shown here, how to remove the nuclei and how to put them in without actually penetrating the egg membrane. And you can see that in this particular experiment, the nuclei are already removed, but the egg membrane is still connected. It is kind of using vacuum to suck plasma membrane, cytoplasm, and nuclei out, and then the other nucleus is put in, and using Sendai virus technology, it's fused with the egg. And I want to show you the movie so you just get a feeling of it. It's very early days. This is analog world. NBC came to film that after we published this paper, and we very soon discovered that when you put this giant camera on top of our microscope, we cannot prevent it sinking. So I say, okay, Jim, you are faster, so you will do manipulations, and I'm stronger. I will stand behind you and hold this camera up so it doesn't sink. And so I apologize, but I deliberately kept this movie just so that you get a feeling what we were trying to do. So this is going through this membrane, which is outside of the egg. You can see it here, zona pellucida, and now we are trying to suck the two pronuclei. And now they are removed, and then you take another pronucleus and a Sendai virus and put it under the zona pellucida. Why are you doing that? Forget it. And what did we want to use this technique for? Well, for many things, and one of them, which we thought that would be very easy to use, is the problem of mammalian parthenogenesis, actually problem of absence of mammalian parthenogenesis. So parthenogenesis is a very good method of reproduction for many creatures, up to and including birds, but it was never possible to do in mammals, despite the fact that we can easily make a normal diploidized eggs, which have all the necessary genes, and they should develop fine, and then for some reason they do not. And there were two possible options to explaining that. One was that sperm brings something functional into the egg which cannot be replaced, and the other one was that we reveal the presence of lethal genes. So we thought we are going to bypass both of these problems, and therefore we are going to have mice made entirely from the female genome or from the male genome by experiment like this, that you take egg, this is now 2 pronuclei, remove the paternal pronucleus and replace it with a maternal pronucleus, and now you have egg with 2 maternal pronuclei. And if you design the experiment properly, you will be able to distinguish between those mice, those embryos, which have a now only paternal genome or maternal genome, and control embryos and all those embryos in which we made a mistake from identifying, because normally mouse pronuclei do not have these little arrows and crosses. So the experimental embryos would have well, you see, that's a problem. You either can show things or slide will advance by themselves. So the experimental one would be agouti mice because they are nuclei from, forget it, I'm not going to touch anything anymore. So the experimental androgenes and gynogenes will come out as agouti mice, and controls will come out as a white mice or black mice. And so all what you have to do is do series of nuclear transfer, put these mice back in, put the embryos back in, and then wait for agouti mice. And the problem is that doing that for, I would guess, probably a year and a half, we could never get agouti mouse. And we couldn't believe that this is so. And we thought, these are perfectly fine mice. Now they are zygotes. They have been fertilized. They have two different genomes, so they cannot be lethal genes, and they are still not going to get born. So after doing many of those experiments, we finally convinced ourselves that it really does seem that you have to have a male and female genome. And the same type of experiment and same manipulations were done by my friend Azim, who will talk afterwards. And the final conclusion was basically that either these things suggest that these genomes are differently imprinted, or the way we put it is that you need both of these genomes because they are functionally different. And we assumed, though we didn't know for sure, obviously, that this is because there are some genes which are expressed only from male and some only from female. The mice, the androgenetic and gynogenetic embryos do not die the same way. There is a, let's call it, phenotype of death of androgenetic and parthenogenetic embryos. This is definitely work of Azim's group. So in the first column, it's normal embryo. Then in column B is the embryo which is made from only 2 female genomes. And as you can see, the embryo is sort of normal but small, but the trophoblast and yolk sac are very much underdeveloped, and probably the embryo is small because there is nothing to feed it. On the other hand, these in C, they are androgenetic embryos. The embryo is truly abnormal and doesn't develop any further, but for example, trophoblast is much more outgrowth. And that was the conclusion in this paper. And it's interesting that nature made a similar experiment in people. Obviously, with people, we cannot transfer nuclei between the eggs and put them back in, but nature did it for us. So in ovary, you can have parthenogenetic activation of an egg, and then you will get ovarian teratoma, which you see to the left of you, I guess. You see the bunch of hair and skin and teeth and bones, and so this is typical structure of benign ovarian teratoma, which is made from only female embryo, so this is only somatic derivations, and on the right-hand side you see the consequence of if a sort of nucleus-less egg is fertilized by 2 sperm, and we know that this is what happens. You get on the lower side, this is hydatiform mole, which happens in this situation, so this is derivative of the chorionic membranes, and on the top, unfortunately, sometimes this hydatiform mole becomes in choriocarcinoma very malignant and bad tumor. So that was the state of the, let's say, imprinting at that moment. And now I will ask one experiment which answers a question: are these cells, the cells which have only male or only female genome, is that the problem which is cell autonomous, or it is the problem which is a structural problem? And this is a very common question in mammalian development, which we often ask, namely, if because of some mutation, for example, the organism doesn't develop liver, is this because the cell doesn't know how to make a liver, or the organism doesn't know how to make a liver? So this experiment was done by Jamie Thompson, who was graduate student in my lab. I guess you might know this name because he later became much more famous for being the first person deriving human embryonic stem cells. But he did this type of experiment. So you start with three kinds of experiment. On the left are gynogenetic embryos, in the middle are parthenogenetic one, and on the right androgenetic. And these embryos are labeled in a specific way. You will see later how. Now we take an individual cell from HL stage embryo and combine it with a normal embryo. And what did we achieve by that? We know that normal embryo will provide all the necessary structures. And so we are now asking, will the cell from manipulated embryo, gynogenetic or androgenetic, will it contribute to all the parts of the embryo, which means that the defect is not cell autonomous, because when we provided the structure, the cell could do it, or actually these cells will not be able to colonize certain part of the embryo. Okay, let's see how it works. So this is the embryo in which we are going to look at. And first we look at the controls to see whether they can contribute to everything. And there you see all these labeled cells, and the reason why they are labeled with these little brown dots is because they have a very large transgene of multiple copies of human beta globin gene in it, so we can do DNA hybridization and find every cell. And when you look at 10 embryos, and the code for this slide is that if it's very red, like brick, that means the contribution from 50% up, and when there is this very faint pink, that means there was no contribution. And you can see that, well, this is due to the geometry of a mouse embryo and where the cell was landing. You can see that in some cases, on the left, the cells contributed only to the trophoblast, then in some cases only to the embryo proper, but in many cases they contribute to everything. And the contribution of the marked cell was basically present in every tissue. Now, what happens in the gynogenetic embryos? We go back. Now you can see the structure. This structure, I really would love to point to things. This structure is ectoderm, this is mesoderm, and this is embryonic part. They are labeled, but extra-embryonic part, which is here, is not labeled. And this is confirmed here. All the contribution of gynogenetic or parthenogenetic embryo is in embryo proper. And that is the part which we saw that is developed normally. Now we ask what happens with the androgenetic embryos, those which look like cannot make a real embryo but can contribute to the trophoblast. We will again examine this stage, and now you can see it's really beautiful. This is extraembryonic ectoderm, which is derivative of trophectoderm, and next to it is a visceral endoderm, and as you can see, it's completely negative. And when you look here, you will see that all the contribution was in extraembryonic membrane. And even more interestingly, in these two embryos, 9 and 10, when the contribution of the androgenetic cell was very strong, the embryo is failing. So it's obviously when due to the geometry of the embryo, these things end up in the wrong place, the embryo is going to fail. Okay, now, as I thought, or I didn't tell you, I sort of hinted, when we did these nuclear transfer experiments, we assumed that the problem is the activation of genes only from the paternal or from the maternal genome. But obviously we didn't have any real evidence for that, though we had some kind of hint. Because the problem is, you see that in these nuclear transfer experiments, you don't transfer genome. It's impossible to transfer genome. You transfer pronucleus, and who knows what part of the pronucleus, maybe some nuclear proteins, maybe some nuclear membrane, maybe there is something truly unique to the male and female pronucleus which we didn't transfer. But we have a hint that the genes were involved by the experiment which we did at that time, and this is using so-called reciprocal crosses. In reciprocal crosses, using this somewhat unusual gene called T-hairpin, and you can see that in cross, in which T-hairpin is delivered by the male, the resulting progeny, so the father is T-hairpin heterozygous, then half of the progeny has a short hairpin tail, and half is normal, as normal standard genetics. When the T-hairpin is delivered by the mother, so the mother is heterozygous, then what we see is that we lose about half of the embryo, and all those embryos which are not lost were actually genetically normal. So the problem with reciprocal crosses is that you could never be sure that the problems, that the phenotype you see is due to the genetics, because as you can see in these two crosses, there are other differences. So in one case, the mother is completely normal, and in another case, she is heterozygous. So you can think, well, maybe the problem is something in the egg cytoplasm which is caused by this gene and not in genes. So you have to distinguish whether we are having problem in the nucleus or in the cytoplasm. And for that, we then applied nuclear transfer. We transferred either paternal pronucleus or maternal pronucleus into a normal egg, and then ask what will be the outcome. And as you can see, following the transfer in these embryos here, they are genetically absolutely identical. They are T-hairpin heterozygous. And the only difference is where the T-hairpin came from. And we were able to show that if T-hairpin comes from the father, we will get short tail, and if it comes from the mother, we will lose the half of the embryos, or practically all of the embryos. So that was firm evidence that maternal T-hairpin lethality is a nuclear and not cytoplasmic effect. And when we were doing these nuclear transfer experiments and getting these, let's call it negative results, we could never get androgenetic and gynogenetic mouse. Jim and I were discussing that and saying, well, it is probably genes, but we don't know whether they are genes, so maybe we should keep on somehow working or waiting until the genes are found. And fortunately, we decided that it's not a good idea. So we published it in early 80s, and the first imprinted gene was found in early 90s, 10 years later, and it was a gene actually in the same region when we, on the basis of T. hairpin experiments, suggested that that's where the genes are. After first imprinted genes was found, then flood opened, and today this is the mouse genome, chromosome map. All these blue things are the paternally imprinted genes. All these red names are maternally imprinted genes. You might notice that they like to come together. They are regulated in many, many possible ways. That is, imprinting is a real baroque of genetics, and complexity of it is truly enormous, and I hope you will be interested enough, or I don't think neither Azim will talk much more about how they are regulated. It's way too long a story. But now I think I will jump into real presence. Very recently, maybe 3 weeks ago, I heard on the radio that people say that one group in China succeeded of making embryos and mice whose genome was derived only from males or only from females, and maybe this showed that males are no longer necessary. And I was thinking, oh, wow, I'm really lucky that we got this prize, because now imprinting is becoming irrelevant. And then I read the paper. So this is the title of the paper, is in press in Stem Cell. And what did they show? They showed, interestingly, unfortunately, imprinting is not lost. What basically show that you can make a pseudo-imprinted genome, that you can manipulate the genome in such a way that it sort of is almost like maternally imprinted, though it was male genome, and paternally imprinted, though it was actually female genome. So what they did is they took so-called haploid ES cells, embryonic stem cells. These haploid embryonic stem cells are a miracle by themselves, but nevertheless, what is interesting about them that as you culture them, they lose methyl group on the DNA. And methylation is one of really essential features of imprinting. Cannot go into it. But the idea was that as they lose methylation, they would lose imprints. And then they combine this by manipulating the genome, by removing some of the genes which they didn't want. So to make a bimaternal mice, they combined the egg. That means that is half of the genome is a normal female imprinted genome, and they used nucleus from this haploid ES cell, which has been demethylated and which also had a couple of genes removed. And they put this nucleus into the egg, and they actually get normal mice. So they succeeded of making a pseudo-male imprinted haploid genome. And it seems that this worked. When they tried to make bipaternal mice, now this is a little bit trickier, because you have to take an egg, remove its genome, then put in one sperm. So this is now normal human imprinted genome, and then you use haploid ES cell, which have been derived from the androgenetically manipulated mouse. And from there, they removed actually many more genes, and they made this construct. So now you have an egg from somebody, sperm and the haploid genome from ES cells, and they make this what they call bipaternal mice, but bipaternal mice were mostly lost, and even one or two which were born alive died immediately. So what this experiment shows is that you can make a pseudo-male imprinted genome and pseudo-female imprinted genome, for the first thing we knew from before, and also that it's much easier to make a pseudo-male imprinted gene, because if you look in here, these are, go back, these are all these, this is several, where it say BM and BM, these are the embryos and adults, so-called bimaternal. And this is the level of methylation. As you can see, all these genes are completely methylated in oocytes. They are not methylated in sperm. And now when you combine that in normal life, you would get about half or 50% methylation, because the imprinting persists. And as you can see, these bimaternal embryos, they actually do express 50% methylation level. When you try to make a pseudo-female genome, that's on the right-hand side, this didn't actually work at all. The bipaternal BP15 embryo remains completely unmethylated. There is no contribution of the pseudo-maternal genome to the methylation, and that's probably why it didn't work. So in true, what this paper shows is that imprinting is very important, absolutely necessary, and that you can fiddle with it in some degree. And then I realized actually none of this is at all necessary if you want to make strange embryos and you want to do strange reproduction. So if me and another gentleman would like to have our baby, what we truly need is somebody to give us an egg. Because what we can do today is we can take our cells, then turn them into ES cell, or what we will be correctly say, embryonic stem cell, induced pluripotent stem cell. And then today we are on the verge of being able to make germ cells out of iPS cells, and Azim will probably talk some about it. And that means that if two men want to have their genetic baby, then they can make their iPS cells, and then from one of them you derive egg, and from other you derive sperm, you combine them, and as long as you find some willing recipient, you will have a completely normal baby, because in this process of going from iPS cells to the germ cells, the imprinting is absolutely correctly established. So actually there is no need to fiddle with imprinting, we can just make our germ cells. And this leaves a little ethical problem, and that is, since we can make germ cells, actually you can make egg, I could theoretically make eggs and sperm from iPS cells derived from myself. I can make both, which means I can make unlimited amount of germ cells and unlimited amount of embryos. And now there comes the question, are these embryos protected, or are they cells in cultures and you can do anything you want with them? So ponder on that. And now to finish, where is the imprinting now? there are these 6 questions which were asked for anything from antiquity. I guess everything starts with Aristotle, as I can keep on seeing, but Rudyard Kipling made a much better song out of it. 4 of these questions, the green one, I would say if we are talking about imprinting, that's what, where during gametogenesis, when during gametogenesis. The problem is that 2 really crucial questions, namely, how does the imprinting mechanism recognize genes during early gametogenesis, at which time all the imprints are supposedly erased? How does this mechanism recognize which genes should be marked to become later methylated and therefore imprinted? And even more important question is why mammals needed to do that? What is the purpose of, because imprinting is evolutionary bad. You have only, instead of having 2 copies of every gene, so you can afford to lose one or have mutation in one and so on, you have only 1 active copy, which is evolutionary bad decision. However, all mammals have imprinting and there is no development without imprinting, so there must have been a very strong reason and evolutionary pressure for it to develop. And probably because it's so recent, it's also so baroque that we don't have a 1 mechanism of imprinting, but multiple mechanisms. But these 2 questions, how and why, will probably keep us busy for some time. Thank you very much.